Welcome back to chapter 14. Now we're into part 2, how these Mendelian genetics prediction, shall we say, algorithms work. It's quite easy to do single trait analysis, but as we understand that traits are often driven by more than one gene, or in the case we may have multiple traits that we may want to assess together, we're going to look at what we call dihybrid crossing as well, which is more complicated. It still uses the same simple rules, but now we have multiple balls in the air like a juggler. So we're going to work through that today, go through a little more terminology, and hopefully get you through with some success. Again, we're going to be using these terms in our discussion today, so it's a good idea to make sure you have a conversational understanding of what these specific terms mean. Also, to remind you that it's a good idea to keep the central dogma in mind, that we have DNA to RNA to protein. And that's what drives phenotype. That is to say, the traits that we observe. We have a specific locus where a gene may or may not be found, or a different form of the gene may be found. In this case, one allele from the one parent has at the locus, it has a functional gene that will synthesize purple pigment, or as part of the metabolic process of making purple pigment. On the other allele from the other parent, this gene is a nonsense mutation, so the enzyme is never synthesized. And so if the plants only had that allele, they would be white because they would not be able to synthesize the purple pigment. So again, this understanding and linking of the genotype to phenotype is rooted in what we call the central dogma, which is DNA to RNA to protein. For most organisms, and this is not entirely true for plants and certain types of hybrids and so forth, plants are much more complicated biochemically. But if we stick to mammalian situations, we have two alleles that we inherit from our parents. If those two alleles are identical, we call that a homozygotic situation, quote unquote true breeding, which is really a bad misnomer. If you have different alleles, then you would be heterozygous for that gene. So heterozygous, then we fall into the idea of whether the trait that is conferred by that allele is dominant or is recessive. That is to say, if you are a heterozygote and you contain a dominant and a recessive allele, the dominant would manifest. The only way that you would see a recessive trait or a recessive phenotype would be that if you have only recessive alleles from your parents. It gets a little sticky after that because we do have codominance and we have incomplete penetrance and some other terms that get at the in-between of some of these things. So dominant versus recessive, it's good to start with a sort of binary situation. That is to say, it's an either or, but in reality, most of our traits are multigenetic. And therefore you get these sort of codominance, incomplete dominance phenomena in reality. In the case of our pea color, our flower color for Mendel's peas, it was fortunate for him that it was sort of a binary situation. You either had the purple pigment or you didn't. So that would be an example of complete dominance versus, say, an incomplete or a codominance. One of the laws that has been corroborated again and again and again by multiple methods that Gregor Mendel originally identified was this idea of segregation. That is to say, only one gene from one parent shows up in the gametes. Okay. Now, this is a simplistic meiosis representation because remember, prior to meiosis 1, we do get this crossing over event and we get this sort of hybrid chromosomes. But let's just for a moment pretend that there's only one gene on these parental chromosomes. So even if we did cross it over, one of these sister chromatids would either be red or blue. But let's pretend that that doesn't happen. Okay, so we get that duplication, and then in meiosis 1, the duplicated chromosomes separate. All right. Then during meiosis 2, the chromatids separate, and we end up with one chromosome from one parent and one chromosome from another parent in the gametes, but not both. Okay, I hope that doesn't confuse you. If it does, please let me know. I'll be glad to work this out with you. Okay, but we're only going to, by the law of segregation says that only one allele will be present in any given gamete, either a maternal allele or a paternal allele. So meiosis ensures that segregation occurs. Okay, even if you have lots and lots of different genes on a given chromosome and they cross over and so forth, at the end of the day, the chromosome down here is only going to have one allele from one parent. Only gets one of the two possible alleles that are present in the parent organism. So let's start with an experiment. Okay, we're going to cross two homozygous parents. When we cross the two homozygous parents, remember that the outcome here is phenotype. We do not know the genotype going into this experiment. When we inbreed purple flowers and purple flowers, so we only get purple flowers all the time. So we know that it is a true breeding parent because it only will give purple flowers when crossed with purple flowers. Same thing's true with the non-purple flowers, the white flowers. We inbreed them for a period of time so that we only see white no matter what. So we know from the reference point that these are homozygous. When we breed two differing homozygous parents that have two different alleles, okay, capital P, little p, little p, in the F1 generation, no matter how many times we breed those two plants, no matter how many offspring we look at, what we see is that only purple flowers exist. That's because even if they are not homozygous, we nevertheless will see purple. Okay, we will always see purple because purple is the dominant phenotype in this case. There are no non-purple flowers because there are none that have only the recessive trait, the recessive gene. However, in the second cross, where we start crossing F1s, okay, we're not using the original parents. If we cross some F1s, and we look at all of the offspring from those F1 crosses, we see a 3-to-1 ratio of purple to white. So we start seeing white flowers again, but it's a 3-to-1 ratio. Well, how does that happen? Well, it can be explained quite easily using Punnett squares. So Punnett square is our tool for understanding why we would see a 3-to-1 ratio in the F2 generation. The real secret is that we can begin to sort of tease out the genetics by looking at the phenotypes first. Rather than knowing what the genotypes are going in, by looking at the phenotypes, we can sort of reverse engineer in some cases, especially in diseases where there are only one or two genes involved, family trees and so forth. We can begin to tease out what the genetics are without knowing the genes, just looking at phenotype. I'm never going to ask you to sort of reverse engineer these things from ratios. What I'm going to do more often is just ask you to calculate ratios of phenotype and so forth. But it's good to know that the reverse engineering is possible and very, very frequently used when tracing family lineage and so forth. Now, I'm guessing most of you at one time or another, if you've had a fair amount of high school biology or perhaps some pre-college courses, have done a basic one-trait Punnett square. It's a very, very simple process. You put the letters of the alleles of the parent over each of the squares, and then you simply combine them. Very, very simple. Like, for instance, here we have a capital A and a capital A, so we would write AA in here. We have a little a and a capital A. Now, by convention, we always write the dominant gene first. And dominants are displayed as capital letters. So even though we have a little letter in some cases, we're always going to write the capital letter first. So capital A for this square, capital A, little a for this square. Capital A for this square, capital A, little a for this square. It gets a little trickier when we get into two hybrid crosses, or if, for instance, we happen to have two heterozygous parents. Okay, so we'll show you that in class so that you can understand how to sort of sort these things without having to write them several times. If we do a homozygous cross where we have a mom with dominant alleles only and a dad with recessive alleles only, one of the patterns that we'll end up seeing, as you see if you do the crosses here we have the little p over here. So, because it's recessive, we write it second. So big P, little p, big P, little p, big P, little p, and so forth. In this case, because we have a double dominant and a double recessive, all of the F1 offspring are going to be heterozygous, but they will all have the dominant phenotype. So there is literally a 0% chance, or near zero, because there's always more complicated than the prediction, a near zero chance of any recessive trait showing up in the offspring of this cross. So normal pigmentation, recessive albino pigmentation, there's a 0 chance because, again, you need to have two lowercase p's in order to have that recessive trait. So virtually 0. There's always exceptions, but in this case we can be fairly confident we're not going to see any recessive traits in the offspring of this cross. Let's just do this cross working it out, just in case it didn't make sense in the previous slide. So we're going to cross two homozygous pea plants, one for the dominant trait of purple and one for the recessive trait of white. And so phenotypically, they're going to be entirely purple. But genotypically, because half of the gametes are coming from capital P and half of the gametes are going to be from the other P, they are all going to be heterozygous, because you're always inheriting a capital P from one parent and you're always inheriting a lowercase p from the other parent. But in the F2 generation, when we cross F1s, we are going to get a different pattern. We're going to get a 3-to-1 purple ratio, where we have one of the four are going to be homozygous for white, one of the four will be homozygous for purple, and two of the four will be heterozygous. So if we had, let's say we had 1,600 offspring. So we had a huge number of offspring. Approximately 400 would be homozygous, approximately 800 would be heterozygous, and another 400 would be homozygous for the recessive trait. Phenotypically, it would be 3-to-1, but genotypically, it's a 1-to-2-to-1 ratio. And Punnett squares will allow you to not only assess via phenotype, it will allow you to predict how many genes or what genes came from where. Again, what we call a sort of a reverse engineering and figuring out where family lineage and so forth may have arisen when it comes to certain types of disorders or diseases or traits. So one-trait Punnett squares are like tossing a single ball in the air. It's a very, very simple exercise. Well, what happens when we start looking at different traits simultaneously? Then the complexity goes up quite a bit. If you remember in the previous Punnett square, there were basically 4 squares. If we do 2 traits, then we have to have 4 times 4, which means that just by adding one more trait, we now have 16 squares. Now the next law that Gregor Mendel sort of pioneered was this idea of independent assortment. Because we have shuffling of genes during sexual reproduction, there are basically 2 outcomes that can happen if you have 2 genes. Either you have 2 genes that are sort of linked, that is to say, when shuffling occurs, they almost always travel together. So a given set of alleles will almost always travel together. And when you look at the deeper molecular biology of it, it has to do with loci, how close together they are on the chromosome. We don't have time to really go into that level of prediction, but you can have genes that sort together, and then you can have genes that sort independently of each other. They're far enough away from each other, or maybe they're on different chromosomes, that we get what we call independent assortment. If they're close together and they're on the same chromosome, the odds of of independence drop significantly. But if they're on different chromosomes or they're way far apart on one chromosome, they're going to sort independently. So let's kind of tackle that a little bit. The way we can sort of determine whether they're dependent or independent is by doing the dihybrid crossing. Okay, we're going to look at a dihybrid crossing and looking at the patterns that generate both genetically and phenotypically. With dihybrid crosses, you can determine whether we have dependent or independent assortment. Now, as you're watching this, I will say that we are going to spend a little bit of time in class going through how to do dihybrid crosses. In this experiment, we're going to start with homozygous for both traits. Capital Y, capital Y, capital R, and then lowercase, lowercase. And so what are the possible gametes that arise from each parent? Well, this simplifies it a little bit because you can only get a capital letter from one parent, and you can only get a lowercase from the other parent. So when you do the cross, you're going to end up with heterozygotes across the board. They're all going to be capital Y, capital R, little r. And they're going to manifest as dominant traits for both of those traits. So there's no way to determine assortment in the F1 generation. It's the F2 generation where we can determine whether there is a dependent or independent assortment. Okay? If we have dependent assortment, it's basically as if both alleles are in the same region, same loci. We get that 3-to-1 phenotypic ratio. Okay, it's just as if the Y and R are one gene, and I don't want you to presume that, but the behavior of the phenotype resembles a single gene assortment. Okay, if we only had Y and little y, we would see that 3-to-1 for Y and little y, there where you have 3 showing the dominant and then the 1 showing the recessive. Okay. The behavior is such that we would only see a 3-to-1 ratio. 3 would be yellow and non-wrinkled, and 1 would be green and wrinkled. Okay. If there is independent assortment, then the odds ramp up significantly. We start to get this 9-to-3 to 3-to-1 phenotypic ratio. And we need to do this in class. It is really, really difficult to show you this in a PowerPoint video. I am going to give you a link to a wonderful resource, the Amoeba Sisters, who do a really good job of showing you how to do this. And we're also going to go over this in class if you would like. The first thing you'll notice about the difference between these two is that we are getting phenotype, we're getting more than one yellow phenotype. We're getting non-wrinkled and wrinkled yellow phenotypes, for instance. We're also getting non-wrinkled and wrinkled green phenotypes. Over here, you are either yellow and non-wrinkled or green and wrinkled, but we don't have that here. Okay. We have, and secondly, we see this again, 9-to-3 to 3-to-1. Okay. 9 of this out of the 16, and again, that's what's the ratio on that? 16% is, or 1/16 is approximately, if I do my little math here, 1 divided by 16 is roughly 6.25%. 0.0625. If you multiply that by 100 to get the percentage, it's 6.25%. So what's 9 times 6.25%? Is equal to about 56%. So a little more than half are going to be round and yellow. About a quarter, a little less than a quarter, are going to be round and green. A little less than a quarter, you see 1, 2, 3, are going to be round and yellow. And then lastly, about 6% are going to be green and wrinkled. That ratio, seeing that 9 to 3 to 3 to 1, would imply independent assortment. All right. And again, we're not going to ask you to reverse engineer too much, but just be aware of it. You see the 3 to 1, you're going to assume dependent assortment, that the genes move together. If you're going to see a more complicated assortment, then you can then imply that it's independent. Just that simple. We are going to show you how to achieve this moving forward, doing those crosses in class, or you can watch the video. But again, understanding why these are different is due to the fact that the genes are either sorting dependently or sorting independently. 3 to 9 to 3 to 3 to 1. All right. Now, the reality is that it never quite works this perfectly. Gregor Mendel was a genius. He was ahead of his time. Many agricultural breeders and so forth had a sense of what was going on, but they didn't really quantitate it like Gregor Mendel did. But Gregor Mendel actually fudged his numbers a little bit. He saw the general trend and he sort of idealized his numbers a little bit. Nevertheless, he was a genius in the ability to understand what was actually happening, even though his data was less than perfect. As I mentioned in previous slides, yes, we are going to be working on dihybrid crosses in class pretty soon. But if you would like a little additional help, I think this video does a wonderful job of showing you the method of doing dihybrid crosses. So if you need a little extra assistance, for instance, go to the cell lab exercises that we have in our department, but also please do look at this video and grab yourself a 16 grid Punnett square. In addition to the method of doing the dihybrid crosses, the Amoeba sisters do a really good job of explaining some of the concepts of dependent and independent assortment. So I think this is a lovely video to go along with what we're talking about in this video or in our class exercises. And a quick reminder that when we have recessive alleles, they cannot be covered up by any other gene that would cause a dominant trait to occur, a dominant allele. Again, very rarely, or I wouldn't say very rarely, but rarely is any trait due to exactly one gene. And I'm going to talk about this in a little bit. But if we have a gene or a set of genes that will cover up the recessive trait, then we will manifest as that dominant trait. So only a person that is homozygous for the recessive allele can manifest the recessive trait. Only show up in individuals homozygous. Now, where this really becomes huge is when we talk about genetic disorders. I mean, particularly what we call autosomal recessive disorders. Now, sex-linked recessive disorders still work, but sex-linked because you're only inheriting, either an X or a Y from your parent. The manifestation is a little bit different there. It still obeys these laws that we see in the Punnett square and Gregor Mendel and so forth, but it's just a little bit different. Okay. Recessive only shows up when you're homozygous for the recessive allele. Folks that are heterozygous for the dominant and recessive, when it comes, particularly when we're talking about autosomal recessive disorders, I guess you could say sex-linked as well, but again, it's a little trickier because of the X and Y situation. If you are heterozygous, you are known as a carrier. So, for instance, one of the most common autosomal recessive disorders for things like hemophilia or cystic fibrosis, we call the parents that are heterozygous, we call them carriers. Another interesting fact about autosomal recessive disorders is the fact that if you have what we call consanguineous relationships, yes, you can think of the very, very ugly term incest, for instance. We do see increased rates of autosomal recessive disorders. We see increased rates in autosomal recessive disorders where we see families having consanguineous relationships. This was never more manifest than in periods of time where royalty only had relationships or married royalty. So there are a number of times in royal history in different royal families where things like leukemia started showing up, okay, or not leukemia, hemophilia, excuse me. Hemophilia showed up, or in the case of for something like cystic fibrosis or other unusual manifestations which would be attributed to autosomal recessive. So this concept that royal genes are superior, that eugenic myth, is actually manifested by the fact that many royal families had these autosomal recessive disorders. Now, to be perfectly honest with you, incestual relationships that are, shall we say, few and far in between, where there's criminality in a family that's sort of spontaneous, if you will, the risk of an autosomal recessive disorder is, while higher in that situation, nevertheless, most children of incestual relationships are just fine. It's just that there's an increased risk. That's one of the reasons why there are specific laws determining how closely related you can be and legally marry because of the risk. And we have figured out now that it's like second or third cousins, something along those lines, not really an increased risk to any significant degree. But one of the reasons why incestual and consanguineous relationships have been outlawed is the fact that there are increased risk of these autosomal recessive disorders. And I've been talking about this for a little while through these slides, but here let's actually look at what we call incomplete dominance, or sometimes we refer to things as co-dominance in some cases. Again, it gets a little more, a lot more complicated, I guess I should say. For instance, we have brown eyes, but not every brown eye is the same shade. Or amber eyes, again, not the same shade. Or green and hazel eyes, not the same shade. Or green to blue, or different shades of blue. Like, for instance, I have blue eyes, but my blue eyes are different from my dad's blue eyes. My mother has green eyes, my dad has blue eyes, and my eyes are much more gray than my dad's eyes. My dad has strong blue eyes. My sister has strong blue eyes as well. So my sister has eyes very similar to my dad, where mine are much more gray relative to both my dad and my sister. And that's because that genes are not completely dominant, not completely recessive. When a gene has more than two alleles, that's where we begin to see some of this. We can get some sort of co-dominance or incomplete dominant patterns. And in advanced genetics, we can actually begin to assess what we call degrees of dominance, where we put a dominant trait. If it is a very strong dominant trait, we would swing it far to the right, manifesting far to the right. And if it's a very strong recessive trait, it is on the far left. So this is a single axis for how dominant or how recessive the trait may be. And so we can get this sort of partial dominance. We get no dominance if it's right in the middle. Okay, a wonderful example of this is actually in skin tone. If we look at somebody who has a highly melanotic parent and a highly non-melanotic parent, oftentimes those children are an in-between color. They are not very darkly pigment. They are a middle, pigment, if you will. Same thing is true with hairstyles and also eye color and other things. We can also get this phenomenon of overdominance and some other things there. Again, it has to do with how the genes are interacting with what's being made in terms of the central dogma, what proteins are made, how those genes are expressed. Again, it gets much more complicated, but we are complicated genetically. And if we're trying to tease these things out, we're just trying to understand the underlying phenomena. So incomplete dominance, codominance are all the reality when we're talking about, especially when we're talking about multiple gene driven traits. Let's look at this phenomenon from the direction of a Punnett square. We are going to assume that white in this case is also a dominant trait. We're assuming it just for purposes here. All right. We also have a dominant red trait as well. So we get a C, superscript R for the dominant red and a C superscript W for the dominant white. And so the F1 generation, the first cross, would receive a CR and a CW, both being dominant, and thus the phenotype is pink. So we have a codominant situation here. Okay, we have a codominant pink phenotype because we're getting half of the red product from one allele and half white product from the other allele. When we look at the second generation, the F2 generation, what we see now is this 1 to 2 to 1 ratio, where we have all, dark red, one of the four, 25%, two pink, 50% will be pink, heterozygous, and then lastly, one of the four, 25% being white. So again, we can reverse engineer and determine whether it's codominant or incomplete dominance based on the pattern that we see across these two generations. So there you have the Punnett square and sort of the relatively straightforward. I know that it doesn't always feel straightforward, but we can, as you work through this, and this is why we have a separate genetics class, is that yes, we can start with a very, very simple one ball, two ball kind of situation, determine whether it's dominant, recessive, codominant, so forth, and we can begin to determine whether it's a single gene situation. Is it a polygenic situation where we have more than one gene influencing something? Very, very common when we're looking at certain traits and behaviors. For instance, diabetes has over 100 genes involved. Okay, it gets really, really complicated. Okay, in fact, it gets so complicated that there's a new theory out there called omnigenic. Rather than saying that there is one gene or multiple genes conferring traits, we get what we say that all genes in the genome are some way or another influencing the behavior or the traits of those genes. And this is pioneered by a guy named Jonathan Pritchard at Stanford. Okay, they have the patterns are getting so complicated, especially for things like I said, over 100 genes for diabetes, that this omnigenic hypothesis has now arisen that all genes in one way or another are influencing traits. We're going to stick to the monogenic and polygenic things in our classes here, in this class, but understand that there is an entire career field when you think about, first of all, genetic counseling for parents who are having issues with children. As I mentioned in class, aside from economics, the number one reason for divorce is actually due to issues with children, where we have rare phenotypes. Okay, so genetic counseling is really important. It's incredibly important in agriculture. Okay, incredibly important in breeding plants and animals for desired traits and or efficiency of calories and so forth. So it's literally a quadrillion-dollar industry. Just to give you a little taste of this, just a little taste. For instance, if we talk about whether your blood is A, B, or O, we can have multiple alleles. So for instance, we can have IA, IB, and then little case I. All right, those are the three alleles. We have two dominant alleles and one recessive allele. When we do the combinations of father and mother, for instance, if she is IA or IB or the gametes of the father, if he's O, he has to have two little i's. You can then start to do the crosses depending on what the gametes are from either the mother or the father. So the IA in the IA allele encodes for the A carbohydrate that gives the A blood type. Remember that carbohydrates are found on the external side of the plasma membrane, with the exception of glycogen and glucose. IB encodes for the B carbohydrate, and the little I encodes for neither. So you have neither the A carbohydrate nor the B. And so again, you can do Punnett squares to make predictions, and we can use those Punnett squares to determine genotype. Comparing phenotype to the possible genotypes gives us how many alleles are involved and whether it's single gene or polygene in terms of the inheritance patterns of the phenotypes. Another type of phenomenon, and this again plays into that sort of omnigenic, it also plays into the multigenic as well. Inheritance and genotypes and so forth is something that we talk about often in endocrinology, and it's called a pleiotropic effect. Because genes code for one or more forms of protein, this is particularly true in immunological situations where you get differential recombination or differential processing of mRNA. We get one or more forms of protein from a given gene. That can lead to one or four or more manifestations or pleiotropy. The issue with pleiotropy is that there is context there. Which cell expresses it, which tissue type expresses it, and how much is expressed. That's the idea of pleiotropy, that we're going to get different effects even though we may have only one gene. All right. Multiple phenotypic effects means multiple effects. Pleiotropy. Pleio is another phrase for plural. Okay, it comes from the sort of the same language or origins. Multiple. Plural means many. Tropy means growth or manifestation. Many manifestations. All right. So a single gene, because you may get different proteins from that gene, we may see differential traits, even though there is only one gene. Here's an example of pleiotropy with a relatively simple genetic profile, and that is cystic fibrosis. All right. We can see pleiotropic symptoms, multiple symptoms based on a relatively simple genotype situation. In the case of cystic fibrosis, you get one allele that's recessive from both parents. It's autosomal recessive. And what do we get? We get manifestations like skin manifestations. You get a salty skin. You get a buildup of salt on the skin. Kind of like if you've been sweating an awful lot and the sweat dries and you get a salty skin. This happens all the time, whether the offspring are active metabolically or not. It just happens even if they're sleeping. They have respiratory problems, frequent lung infections. They don't grow very well, and they have what we call failure to thrive. When they're born, they often have high levels of what's called meconium. This is where the child seems to be under stress and ingesting a lot of amniotic fluid, which leads to a certain type of fecal manifestation. Or after they're born, they have an unusual manifestation of the fecal material, bulky, greasy. Again, that has to do with the multiple manifestations of what this gene is doing in different tissue types. The problem with cystic fibrosis is that cells that are actively secreting chloride can't. And so whether that happens in the skin, happens in the respiratory tract, happens in the digestive tract, or is a sum of all of those traits, that's what we mean by pleiotropic manifestations from a single gene. A bit complicated. Take your time with it and have patience with yourself. Understand what the word origins mean. Plural manifestations, plural growth, if you will. Before we finish lecture today, I've got a couple more slides for you. I know we're going a little bit long than my normal online lectures, but bear with me for just a second. What we've been doing up to this point in this chapter is really just talking about how, genes sort on a sort of macro level. We did talk about, meiosis and so forth. Remember, too, that some of the complexity may be due to the fact that some of the complexity that we're seeing can be corroborated by the DNA replication that we're going to talk about. We've talked about a little bit before, but now in subsequent chapters, we're going to talk about more. So in the next week or two, we're going to be getting into DNA replication further. Remember that with DNA replication, what we have going on is that we're going to separate the parental strands into individual templates. So if we heat the strands, what we say melt the strands. When we get into replication, we talk about melting the strands. It's not solely due to heat. We get the separation of the strands into individual templates, and then we fill in the blanks of the template with new nucleotides. Again, heating and cooling is causing this separation. If we cool the strands, we get a solidified parental double-stranded molecule. If we heat or we add proteins, okay, we can get individual strands. Some of the assortment and some of the issues that we see in this in genotype phenotype have to do with the mechanism of base pairing and template strand. That DNA replication is a somewhat mitogenic event, and that sometimes we get spontaneous changes in phenotype due to the fact that if we have mitosis, there is a risk of an error being introduced. And if we have lots of mitosis, like in the case of inflammation, then the risk of errors increases. So that's one of the mechanisms for why we see increased complexity beyond the lovely Gregor-Mendel predictions. So this completes chapter 14. Please do make sure that you read the chapter in addition to watching this video. I have not, for issues of brevity, I've worked on some of the key things, but make sure you go over the terminology, you read through, and that you do the questions in the chapter, and in class we'll be doing some exercises to work on Mendelian crosses and how Mendelian levels of prediction work. Thank you for your time, and I will see you in class. Goodbye.